Assalamualaikum. I'm going to present on the abstract of this article first. A spatially discrete expression of transcription factors. Spatially means here is the genes that are expressed on specific locations of the tissue. Determines the anterior to posterior organization of the early embryo in Drosophila. Whereas toll pathway and TGF-beta signaling pathway, TGF-beta is a cytokine that involved in many cellular processes. Both of the pathway determines the early dorsal-ventral pattern in Drosophila. Embryonic MAPK or ERK signaling contributes to both anterior to posterior patterning in the terminal region and to dorsal to ventral patterning during oogenesis and embryonic stage of the Drosophila. ERK is actually a member of MAPK family. Here, figure 1 shows the MAPK or ERK pathway that involves many different proteins, activators, transcription factors, and also regulators. Next, we are going to look a little bit on what happens if RAF gene is mutated. First and foremost, it leads to the loss of ventral sulfate through the loss of ventral furrow at the ventral part of the embryo cell. We can see the location of this ventral furrow in figure 2. Next, it will lead to the absence of dorsal or NF-kappa B nuclear localization. This absence of nuclear localization causes no nuclear concentration gradient in the early embryogenesis stage. Hence, no specification of germ layer along the dorsal-ventral axis. Besides that, there will be absence of mesoderm determinants, which are called as TWY and SNA genes that are required in ventral sulfate. Then finally, disruption in TGF-beta signaling pathway. Let's move on to the introduction of this article. So basically, active toll pathway adopt ventral sulfate and cells with active TGF-beta pathway adopt a dorsal sulfate in Drosophila. RAF kinase, as the name says, RAF gene is a type of kinase that catalyzes the transfer of phosphate group from a donor to a substrate. RAF plays many roles in organism development, especially in Drosophila. RAF acts downstream of dorsal receptor for patterning anterior and posterior terminal, meaning that this gene is located downstream of dorsal receptor and it is activated together, leading to the transcriptional activation of zygotic genes. Other than that, it also acts downstream of EGFR to determine ventral, ectodermal, and lateral fates during embryogenesis, which means it is located downstream of EFG receptor to determine the ventral fate of ectoderm in the embryo. Besides that, RAF acts upstream of SENA and downstream of RAS and SAB genes, which means RAS and SAB gene is upstream of RAF, while SENA is downstream of RAF. This sequence is important to activate the R7 photoreceptor cells in the eye. Now we are going to discuss further on how dorsal ventral patterning occurs in Drosophila. Number 1 is embryo. Number 2 is gherkin, that is higher at the dorsal side. Gherkin mRNA will be expressed through gherkin protein. It activates the EGF signal. This activation of gherkin leads to the transcription of rhomboid gene. It is required to induce dorsal sulfate in the oocyte and consequently in the embryo. Number 3 is torpedo receptor. Gherkin will also bind to the torpedo receptor. This activation of both EGF signal and torpedo receptor causing the pipe production to be inhibited, which is at the follicle cells at number 4. Therefore, this pathway leads to dorsal axis. Now we are going to look at the ventral axis formation. Again, number 1 is embryo. Number 2, since there are no gherkin protein at the ventral side, pipe protein will be expressed. Number 3, this results in release of spasal protein. Number 4, spasal then activate toll receptor pathway through the toll receptor. Krepfen bind to toll and interact with tube. Tube participates in many protein-protein interactions. Number 5, tube and pali binds to form a complex. Number 6, tube and pali will phosphorylate the cactus protein and therefore degradation of cactus occurs. And now dorsal is free. Number 7, dorsal will translocate to nucleus to initiate or to repress the expression of specific genes such as TWY, SNA, and ZR, which mostly involve in ventral patterning and the rest of the embryo.
Thank you, Shifa, for the explanation. Now, I will be talking about the methods used in this research article. The first method that was used in this paper is crosses and expression of UAS construct. The image below shows the crosses that were conducted. The maternally mutant eggs were generated by the dominant female sterile technique, where balanced mutants are crossed to the dominant female sterile mutation, that is OVOD2, and recombination is induced using the FLP-FRT method in ovaries, which is a site-directed recombination technology increasingly used to manipulate an organism's DNA under controlled conditions in vivo. Oregon R was used as the wild-type strain, while RAF 926 was the mutant used. As mothers were heterozygous for the Gal4 source, only half of the embryos will express Gal4. Next, I will be talking about mutant, transgene, and driver lines. Before that, I want to define what is transgene and driver lines. For those who are not familiar with this word, transgene is a gene that has been transferred from the genome of one species into another, while driver lines, from the name itself, will drive expression of something. In this paper, the mutation was genetically mapped on the chromosome X by complementation with deletion or duplication strains. The third method used in this paper was immunofluorescence. Embryos were fixed with heat methanol treatment or with heptane or 4% formaldehyde in phosphate buffer at pH 7.4, and the antibodies used were 1/10 or 1/100 antidorsal. Next, this method uses a light microscopy. The embryos at cellularization stage and earlier stages were selected using halocarbon oil. These embryos were then carefully oriented such that the embryo was upright with the anterior-posterior axis aligned with the axis or perpendicular to the axis of the glass capillary. Other than that, a scanning electron microscopy has also been used. The imaging was performed with Hitachi TM3030 plus tabletop scanning electron microscope at 1000. Below shows the image of scanning electron microscopy, which provides a higher resolution images through optimization of the electron optics system. The sixth method used was RNA preparation and RNA sequencing. The extracted RNA was quantified using nanodrop, while the library preparation was performed using 1 microgram of total RNA, and sequencing was performed using Illumina High Sequence 4000 system. During RNA sequence analysis, the rats annotated as rRNA, snRNA, or snRNA were removed. Transfection and protein quantification was also a method used in this research article. In this method, untransfected cells were used as negative control. Other than that, live cell confocal microscopy was also used. The cells were imaged 24 hours after transfection using the ZESS LSM 800 confocal microscope. Cells transfected with either dorsal or ERK were used as negative controls for respective experiments. Lastly, for the data availability, the RNA sequence data from this study has been deposited to GEO for our references.
Next, I'm Nur Shafika, and I'm going to explain about the result. CRISPR-Cas9 approach, where maternally expressed Cas9 drives gRNA-directed mutation in the RAF gene, as the CRISPR-Cas9 system introduced mutations randomly at different developmental time points, we observe a variety of phenotypes including docilized embryos, as shown in the presence of dorsal hair, as in figure A to F, as well as other patterning phenotypes which like to depend on the timing of the induced genetic lesion. RAF 926 showed a similar high docilized phenotype, where the embryos appear to twist around themselves and fail to develop ventral structures, but ovarioles were unaffected, and oocytes developed standard shape and polarity including normal dorsal appendages. It began to show abnormalities during the first process of gastrulation on figure F, where no ventral furrow is formed and the style movement of gastrulation in docilized embryos become unpredictable in figure G and H. This showed a twisting and elongation of ectodermal tissue, as in figure E to H, without the ventral denticle structures that will be seen in ventralizing mutation, such as characters in Figure C. Based on the result, genes that were upregulated in RAF96 mutants were enriched for processes associated with ectoderm, including epithelial cell morphogenesis, whereas the set of downregulated genes was enriched for the processes related to nervous system and mesoderm development. GSEA identified genes involved in embryonic organ morphogenesis as dysregulated in RAF96 embryos. These results are consistent with the lack of ventral furrow formation, as many internal structures will be missing without the generation of a mesoderm in RAF96 mutants. In particular, the DPP pathway regulates SOG and BRK, as well as the neuroectoderm determining transcription factors intermediate neuroblast defective, or IND, and ventral nervous system defective, or VND, who downregulated. Genes that are repressed by dorsal and as such are expressed on the dorsal side of the embryo, which gives rise to the ectoderm. In dorsalized embryos, a condition where no toll signal is present, these genes increase in expression. The transcriptional analysis showed that RAF96 mutants recapitulate the effect of strong loss of function mutants in the toll pathway, with most key mesoderm and neuroectoderm determining genes being significantly downregulated, and ectoderm determinants being significantly de-repressed. This pattern of expression changes supports that these are dorsalized embryos, which lack both the mesoderm and neuroectoderm germ layers, and are essentially empty epidermal pouches. The surprising finding was that despite upregulation of toll signal transduction pathway genes, the expected transcriptional targets of dorsal were downregulated, suggesting RAF is involved in the pathway of functions in parallel to dorsal, but is required for full pathway activity.
Based on the lack of a ventral furrow and the loss of dorsal-induced genes, dorsal-ventral patterning pathways are looked upon to determine the mechanism through which RAF is required for ventral furrow formation. The lack of ventral furrow in RAF926 was like dorsal and toll mutants, so the activity of the toll pathway in mutant embryos is evaluated. The dorsal protein localizes to the nucleus in a dynamic manner in response to toll signaling in the ventral cells of pre-gastrulation embryos. To observe this process in living embryos, dorsal green fluorescent protein (GFP) is used to image the early stages of toll pathway activation. In normal development or the wild type, a gradient of nuclear dorsal GFP can be observed before the ventral furrow forms, and the cells at the center of this gradient invaginate to begin ventral furrow formation. However, in RAF926 embryos, which refer to the pictures in the box, it showed no visible nuclear localization on the ventral side. From Figure 5, ventral furrow cells form the mesoderm after invagination. This process requires the twist and snail genes. Twist is first expressed within the presumptive mesoderm located on the ventral side of the embryo. It acts with dorsal to establish the presumptive mesoderm, and once mesoderm differentiation begins, twist expression is maintained. Twist and snail then drive mesoderm formation. In the transcriptional analysis, it has been observed that the RAF926 mutation showed a significant decrease in the expression of both genes. Since the mesoderm is established by ventral cells, twist in this case acts as a ventral mesoderm marker that gives us insight on how abnormal ventral development in early stages can disrupt late developments of the mesoderm. In a wild-type embryo, the ventral cells develop properly during cellularization and gastrulation, leading to ventral furrow formation and normal mesoderm development. By contrast, RAF926 embryos lack twist GFP signal, showing only yolk autofluorescence. Moving on to Figure 6, a recently developed model of EGF signaling which uses an optogenetically active allele of SOS is implemented. As the major advantage of the opto-SOS is that expression of the allele alone does not affect flies if they are kept in the dark. So expression can be driven maternally in the F2 generation, leading to very early EGF activation. This has allowed Opto-SOS to be used to study patterning, transcription, and morphogenesis. The loss of RAF led to a loss of ventral cell phase and to a loss of twist expression. Activation of RAF through expression of Opto-SOS can also inhibit ventral furrow formation. In contrast to the loss of function condition, Opto-SOS expression activated twist GFP expression in all cells of the ectoderm. These findings suggest that RAF hyperactivation and loss both affect ventral furrow formation. As we can see from the bottom right picture, the loss of RAF leads to formation of single germ layer instead of 3 germ layers.
Today I would like to explain about discussion. Although previous study has comprehensively screened and characterized many RAF mutants, no connection between RAF and ventral fit determination has been shown. Scientists has applied a modern screening technique where a series of mutations have made and characterized through the whole genome sequencing and found a strong mutation in RAF that leads to a complete loss of ventral structure. Scientists discovered that this phenotype was not due to the defective of oogenesis and could not be explained completely by defective dorsal signaling. Secondly, most importantly, this RAF function appears to be independent of the MAPK cascade, as there is no evidence of active MAPK in ventral cells. Studies looking at phosphorylated MAPK show activation at the anterior and posterior poles in the early stage. Interaction between MAPK cascade and toll pathway has been observed at early stage through Capicua and WNTD. This suggests that the RAF function in the most ventral cell is not through MAPK cascade, but could perhaps function through direct phosphorylation of I-kappa beta/cactus. Lastly, there is a limitation of study because although scientists shows that overexpression of truncated RAF fragment tagged with M-cherry. M-cherry is a protein derived from Cnidarius and is used as fluorescent tracer in transgenic or transfection experiment. Even the mutant RAF alleles was tagged with this M-cherry, it still not show any dominant negative effect. It is not possible to rule out completely that there are dominant effects of the truncated RAF alleles due to the possible interference of the M-cherry tag. Further, RAF alleles with M-cherry tagged was expressed in an otherwise wild-type RAF background. And although duplication mapping did not show lethal second site mutation, it remains possible that non-lethal mutation exists on the chromosome that affects the dorsal ventral patterning. Scientists did attempt to expand their studies to the other novel RAF mutants, but these did not produce embryos, suggesting further studies will be required to understand the RAF roles in embryogenesis.